Bismillahirrahmanirrahim. Assalamu alaikum. Our today's topic is about carbohydrates. These are the references and the course contents are structure of carbohydrates, classification of carbohydrates, properties of carbohydrates, biological significance of carbohydrates, chemistry of carbohydrates, describe the structure, properties and function of carbohydrates, and define carbohydrates, describe the general structure of carbohydrates, explain classification of carbohydrates, compare three major classes of carbohydrates, that is monosaccharide, disaccharide and polysaccharides, discuss the biological significance of carbohydrates and the objectives. These are the objectives. Now carbohydrates. Carbohydrates literally mean hydrated carbon. Carbohydrates are composed of carbon, hydrogen and oxygen. The ratio of hydrogen and oxygen is the same as in water. देखिए कार्बोहाइड्रेट का मतलब ये होता है लिटरली कि इसका मतलब ये है कि हाइड्रेट ऑफ कार्बन यानी इसके अंदर कार्बन हाइड्रोजन और ऑक्सीजन होती है और जो हाइड्रोजन और ऑक्सीजन का रेशियो होता है वो 2:1 होता है वैसे ही जैसा कि वाटर के अंदर होता है। कार्बोहाइड्रेट्स पॉलीहाइड्रोक्सी एल्डिहाइड्स होते हैं या फिर पॉलीहाइड्रोक्सी कीटोन्स होते हैं। ठीक है, पॉलीहाइड्रोक्सी एल्डिहाइड ये देखिए ये एल्डिहाइडिक ग्रुप है, सी डबल बॉन्ड ओ और एच एल्डोज शुगर कहलाते हैं और पॉलीहाइड्रोक्सी कीटोन्स जो हैं वो कीटोन्स शुगर कहलाते हैं। कॉम्प्लेक्स सब्सटेंसेस दैट ऑन हाइड्रोलिसिस यील्ड पॉली OH एल्डिहाइड और कीटोन। Some are exceptions. General formula CN H2N H2ON. Carbohydrates with different formula, for example uronic acid C6H9O7 and fucose C6H12O5. Non-carbohydrates with formula of carbohydrates formaldehyde inositol, just formaldehyde CH2O, and this is inositol. डी ग्लूकोज, देखिए ये डी ग्लूकोज का स्ट्रक्चर है, इट इज एल्डोज शुगर। डी ग्लूकोज का फॉर्मूला आप इस तरह से समझ सकते हैं कि थर्ड कार्बन का OH लेफ्ट साइड पर होता है। ठीक है, फिर ऑन ऑक्सीडेशन इट गिव्स डी ग्लूकोरोनिक एसिड। The next is example is D-fucose and L-fucose. D-fucose and L-fucose are the mirror images of each other. ये दोनों एक दूसरे के mirror image हैं। और इसी तरह D-galactose और L-galactose, they both are the mirror images of each other. Examples of carbohydrates: glucose, major metabolic fuel for mammals. Glycogen, storage in animals. Storage in plants are starch. Cellulose, structure provided structure cell wall, it's made up of cellulose. And plants and papers. Chitin, it is it provides structure in arthropods. Ribosomes, RNA, ATP and NAD. Ribose are and deoxyribose, DNA, present in DNA, and lactose, present in milk. It is the structure of lactose. It's a disaccharide. of two monosaccharide, that is galactose and glucose. Here are the structure of carbohydrates. Now comparison of carbohydrate. Sucrose present in plant. Cellulose source plant. Subunit beta glucose. Bonds bond 1,4. Branches no, and its diagram is and its shape. The second is starch types amylose and amylopectin present in plant. uh, Subunit alpha glucose. The subunit of amylose is alpha glucose and 1,4 linkage. Branches no branches. Its structure and shape. The second is amylopectin present in plant, alpha glucose 1,4 and 1,6 linkage. Branches are present approximately per 20 subunits, and its structure and its shape. The third is glycogen present in animal, alpha glucose 1,4 and 1,6 bond. Branches are present approximately per 10 subunits, its structure and its shape. Now the food high in carbohydrates: sweets, soft drinks, breads, beans and peas, cereals, rice, maize, barley, wheat, corn, apricot, dates, blueberry, banana, fig, grapes, apple, orange, pear, pineapple, strawberry, watermelon and raisins, macaroni, spaghetti, potato and carrot. These are the foods high in carbohydrate. Now the function of carbohydrates. Carbohydrates have six major functions within the body. It provides energy and regulation of blood glucose, sparing the use of proteins for energy, breakdown of fatty acids, biological recognition process, flavor and sweeteners, and dietary fiber. Saturated fatty acid, stearic acid, octadecanoic acid. It is adenosine triphosphate, ATP. Carbohydrate is necessary for the regulation of nerve tissues and is the source of energy for the brain. Some carbohydrates are high in fiber, which helps prevent constipation. It is structural components. Carbohydrates are also important for the correct working of our brain, heart, and nervous, digestive, and immune systems. Polysaccharides, storage of energy, example starch and glycogen. Structural components, example cellulose in plants and chitin in arthropods. Ribose is coenzyme, example ATP, FAD and NAD, and the backbone of RNA. Deoxyribose, component of DNA. Heparin is used to treat and prevent blood clots from forming, especially in the lungs and legs. Carbohydrates are monosaccharides, disaccharides, and polysaccharides. Example of monosaccharides are glucose, fructose, and galactose. And example of disaccharides are maltose, sucrose, and lactose. And polysaccharides are starch, glycogen, and cellulose. Carbohydrates are of simple types. Simple carbohydrates are monosaccharides and disaccharides. Monosaccharides contain one sugar unit. Example are glucose, fructose, and galactose. And disaccharides contain two sugar units. Example are lactose, sucrose, and maltose. Complex carbohydrates are oligosaccharides having 3 to 10 sugar units. Example raffinose and stachyose. And polysaccharides uh, having more than 10 sugar units are glycogen, starch, and dietary fibers. Now classification of carbohydrates. Saccharides, physical properties, uh, low molecular weight, soluble in water, sweet to taste. Composition, monosaccharides and disaccharides. Monosaccharides, simple sugars, and disaccharides, double sugars bonded with glycosidic bond. General formula CH2ON and N may be 3 to 7. Common example are glyceraldehyde, glucose, fructose, galactose, and ribose sugar. Disaccharides having C12H22O11. Example are maltose, sucrose, and lactose. Polysaccharides contain complex sugars, high molecular weight, insoluble in water, tasteless. Multiple sugars bond with glycosidic bond linkage. Example starch, glycogen, cellulose, lignin, and chitin. Having general formula CXH2ON. Now monosaccharides. They are simplest, colorless, water soluble, sweet, crystalline solids. They can be classified according to four different ways. Placement of C double bond O group, either it is aldo or keto. Number two, the number of carbon atoms, di, tetra, and penta. Number three, chiral carbon handedness, D or L. Number four, rotation of plane polarized light, D or L. These are the four ways to classify monosaccharides. Classification number one, placement of C double bond O group. Number one, aldoses containing aldehyde group, and number two, ketoses containing ketone group. Number of carbon atoms, diose, glycoaldehyde, triose, glyceraldehyde, dihydroxyacetone, tetrose, erythrose and erythrulose, pentoses, ribose, ribolose, hexoses, glucose, fructose, haptoses, pseudohaptulose and monohaptulose, octoses, methyl thiolinocosamide and nonoses, neuraminic acid and sialic acid. These are the example of aldotriose, which is D-glyceraldehyde, aldotetrose, D-erythrose and D-threose. These are aldoses. Aldopentose, D-ribose and D-arabinose, D-xylose and D-lysose. Hexoses, aldohexoses, D-allose, D-altrose, D-glucose, D-mannose, D-glucose and D-idose. Now ketosis, ketotriose, triolose, dihydroxyacetone, D-erythrulose, ketotetrose, tetrulose, ketopentose called pentulose, D-ribonulose and D-xylulose. And ketohexoses, hexulose, D-cyclose and D-fructose, D-sorbose and D-tagatose. Disaccharides, glycosidic bond, example maltose, having two glucose unit, lactose containing galactose and glucose, sucrose containing glucose and fructose. These are the example of disaccharides bonded with glycosidic bond. Mono and di ends and O's. Mono and di also called sugar. Blood sugar, glucose, table sugar, sucrose, glucose plus fructose, milk sugar, lactose, galactose plus glucose, and malt sugar, maltose contain glucose and glucose, having two glucose unit. Now isomerism. In chemistry, isomers, isos means equal and meros means part, are compound with the same molecular formula but different structural formulas. Number one, structural isomer and stereoisomerism. Stereoisomerism. Same structural formula, different special configuration with respect to the penultimate carbon atom. Glucose has two isomers, D-glucose and L-glucose. It is D-glucose and L-glucose. अगर आप स्टूडेंट्स इसमें नोटिस करें तो डी ग्लूकोज के अंदर थर्ड कार्बन का OH लेफ्ट साइड पर है और बाकी सारे सेकंड, फोर्थ और 5 कार्बन का OH राइट साइड पर है। लेकिन जब आप एल ग्लूकोज को ऑब्जर्व करें तो आप नोटिस करेंगे कि इसके अंदर थर्ड कार्बन का OH राइट साइड पर है और बाकी सेकंड, फोर्थ और 5 कार्बन का OH लेफ्ट साइड पर है। तो दे बोथ आर स्टीरियोआइसोमर्स। Now enantiomers are the mirror images and non-superimposable. enantiomers वो आइसोमर्स होते हैं जो एक दूसरे के मिरर इमेजेस हों, लेकिन अगर हम सुपर इम्पोजेबल ना हो, जैसे अगर हमारे दो हाथों को आप देखें तो दोनों हाथ एक दूसरे के मिरर इमेज हैं, आप उनको आमने सामने करके देखें तो वो एक दूसरे के मिरर इमेज हैं। लेकिन अगर आप एक हाथ को अपने दूसरे हाथ के ऊपर रख कर देखें तो वो उसके ऊपर सुपर इम्पोजेबल नहीं है। बिल्कुल इसी तरह से अगर इस कंपाउंड को मैं उठाकर इस कंपाउंड के ऊपर रख दूं तो ये एग्जैक्ट ऐसा नहीं है, बल्कि ये एक दूसरे के मिरर इमेज है। तो इसीलिए इनको हम कहते हैं इनेंशियोमर्स। नाउ डायस्टीरियोमर्स, नॉन मिरर इमेजेस एंड नॉन सुपर इम्पोसिबल। डायस्टीरियोमर्स वो आइसोमर्स होते हैं जो ना तो एक दूसरे के मिरर इमेज हों और ना ही सुपर इम्पोजेबल हों। अगर आप इस कंपाउंड को देखें तो हाफ कंपाउंड जो है वो सुपर इम्पोजेबल है और हाफ कंपाउंड जो है वो मिरर इमेज है। तो ये एक दूसरे के डायस्टीरियोमर्स हैं। इसी तरह से एपीमर्स, चेंज इन कन्फॉर्मेशन अराउंड ओनली वन कार्बन। These are epimeric carbon. Two D representation of a 3D structure. As we know that molecules have 3D structures, the paper board allow us to draw only 2D structures. We cannot draw 3D structures on a plain paper. Fisher projection for open chain, Netta projection for more accurate representation, and Hayward projection for ring isomers of sugars. Now it is Fisher projection. The Fisher projection is a two-dimensional representation of a three-dimensional organic molecule by projection. Rules: all bonds horizontal or vertical lines. Carbon chain: vertical carbon atoms in center, C1 top. In all aldose, carbon of the aldehyde C1. Ketose, carbon of the ketone lowest number. Horizontal bond towards the weavers, vertical bonds away from the weavers. Howard projection. Howard projection is a common way of representing the cyclic structure of monosaccharides with a simple three-dimensional perspective. It is Fischer projection of D-glucose. It is Howard beta D glucopyranose. It is abbreviated Howard. A Howard projection has the following characteristic: atoms numbered 1 to 6 carbon atom, carbon number 1 anomeric carbon. Atom 1 to 6 have extra hydrogen atoms not depicted. A thicker line closer to the observer, 2 and 3 OH. Atoms 1 and 4 farther from the observer, 5 farthest. Groups below the plane of the ring equivalent to right hand side of a Fischer projection. This cyclic isomers, this alpha D glucose and beta D glucose. These are the anomers alpha and beta. Now reducing sugars. A reducing sugars is any sugar that is capable of acting as a reducing agent because it has a free aldehyde group or a free ketone group. All monosaccharides are reducing sugar along with some disaccharides, some oligosaccharides and some polysaccharides. The monosaccharides can be divided into two groups: the aldoses, which have an aldehyde group, and the ketoses, which have a ketone group. Ketoses must first tautomerize to aldoses before they can act as reducing sugars. Disaccharides are formed from two monosaccharides and can be classified as either reducing or non-reducing. Non-reducing disaccharides like sucrose and terahallose have glycosidic bond between their anomeric carbon and thus cannot convert to an open chain form with an aldehyde group. They are stuck in the cyclic form. Reducing disaccharides like lactose and maltose have only one of their two anomeric carbons involved in the glycosidic bond, while the other is free and can convert to an open chain form with an aldehyde group. Reducing sugar have an aldehyde group in open chain form, monoglucose, fructose, glyceraldehyde, galactose, dilac- dilactose and maltose, di-non-reducing, sucrose and terahellose. Hemi-vis-full acetal and ketal. This hemiacetal, hemiketal, acetal and ketal. These are carbohydrates, aldose and ketose. Physical properties. Mono and disaccharides are crystalline solid. Polysaccharides are amorphous solid. Mono and disaccharides are soluble in water. Polysaccharides are insoluble in water. Mono and disaccharides are sweet in taste. Polysaccharides are tasteless. Mono and disaccharides are low molecular weight. Polysaccharides are high molecular weight. Now, chemical properties. Formation of glycosides. In chemistry, a glycoside is a molecule in which a sugar is bound to another functional group via glycosidic bond. Chemical properties. Ozone formation with phenyl hydrazine. Ozone formation mechanism. Benedict test show positive test for reducing sugar reaction. Reducing sugars are oxidized by the copper ion in solution to form a carboxylic acid and a reddish brown precipitates of copper 1 oxide. Now preparation of Benedict reagent. When copper sulfate react with sodium carbonate in the presence of sodium citrate, Benedict reagent is formed. Benedict test reaction. When Bendix reagent react with aldehyde, carboxylic acid is formed with brick red precipitates of cuprous oxide. Felling test. Felling can be used to distinguish aldehyde with ketone functional group. The compound to be tested is added to the Felling solution and the mixture is heated. Aldehyde are oxidized giving a positive result, but ketones do not react unless they are alpha hydroxy ketone. These tests are useful to check for glucose in the urine of our diabetics. Felling test reaction. Now Felling solution, copper sulfate plus sodium hydroxide plus potassium sodium tetrate, Rocale salt, heat, it form water, sodium sulfate and Felling solution. Felling test give red precipitates of cuprous oxide. It shows the presence of aldehyde. Oxidation, it is also possible to oxidize a monosaccharide to a carboxylic acid. There are two important oxidation of an aldehyde aldose to an aldonic acid, oxidation of the alcohol on the highest numbered carbon atom to a uronic acid. Open chain form of an aldose and aldonic acid. Glucose, for example, glucuronic acid. Ester formation, monosaccharides like all alcohols may react with acid to form esters. Any of the alcohol groups may react. Glycosidic bond formation, monosaccharides plus monosaccharides condense together with the release of water molecule to form glycosidic bond. It is disaccharide maltose. When this disaccharide hydrolyzes, it again form monosaccharide. Bacterial cell wall. The cell wall is a tough, flexible, but sometimes fairly rigid layer that surrounds some types of cell. It is located outside the cell membrane and provides these cells with a structural support and protection in addition to acting as a filtering mechanism. A major function of the cell wall is to act as a pressure vessel preventing overexpansion. When cell wall are found in plant, bacteria, fungi, algae and some archaea, animals and protozoa do not have cell walls. The material in the cell wall varies between species and can also differ depending on cell type and development stage in bacteria. Peptidoglycan forms the cell wall. Archaean cell walls have various composition and may be formed on glycoprotein, S layer and pseudopeptidoglycan or polysaccharides. Fungi possess cell walls made of the glucosamine polymer chitin. Algae typically possess walls made of glycoproteins and polysaccharides. Thank you very much. Allah Hafiz.